All right, so we should get started back up. We're thrilled that Kaisu Lankinen is with us to talk about her recent study using HNN for multisensory integration. Kaisu is a postdoc at Mass General Hospital doing some really interesting MEG work and recently published a paper in the Journal of Neuroscience using HNN. So thank you so much, Kaisu. And it will go for about 20 minutes, about 15-minute talk, and then any questions. I think Kaisu has a great perspective on how to go from nothing to publication with, no, I don't mean nothing. I meant never having used, that came out totally wrong. We're going to delete that in the recording. Having never used HNN or not having a lot of experience with neuromodeling in general. Am I right, Kaisu?
Yes. Yeah, that's right.
Coming up with a study that was publication quality in the Journal of Neuroscience. So congrats on that. It's a recent publication.
Thanks.
So thank you.
Okay. Just a moment. Okay, do you see my slides okay? Okay. Yeah, so thank you so much for inviting me to talk here. So as Stephanie mentioned, I'm a postdoc in Mass General currently in Auditory Cognition Lab by Jyrki Ahveninen. And I'm going to talk about modeling MEG responses in auditory cortex using HNN. So we were specifically focused on investigating differences of sensory specific versus cross-sensory brain activity. In auditory cortex, sensory specific activity refers to activity evoked by auditory stimulus. So it comes through subcortical pathways through thalamus to auditory cortex as feed-forward type input. By cross-sensory activity, we mean activity that is response to another sensory modality. Here, for example, visual stimuli. So the visual information arrives to auditory cortex, presumably as feedback type input. Visual activity has been shown before in auditory cortex with previous MEG and fMRI studies, but here we wanted to get into very details what happens underlying these signals. So it has been shown with animal recordings that these different types of activations have a different type of laminar activity profile within the cortex. So here is a figure from monkey study by Schroeder and Fox, where they measured current source density and multi-unit activity in the auditory cortex. So they found that they used also the same auditory and visual stimuli, and they were looking at the auditory cortex. So they found out that feed-forward type input arrives in the middle cortical layer, and feedback type evoked by visual stimulation arrives into superficial and deep layers. So as Stephanie earlier mentioned, reaching this or studying the animal models before doing your human study provides a very good starting point for your forming the hypothesis. So in our experiment, we wanted to measure these feed-forward and feedback effects with MEG, and we wanted to investigate the neuronal underpinnings of the related processes by using HNN. So in our experiment, we had eight subjects in MEG, and we had two different tasks. So in the noise task, we presented brief noise bursts and or visual checkerboards and their combinations. And then we had a letter task where there was a written or spoken word, like not verb, but a letter, or their combinations. And the tasks for the subject was to react to occasional oddball targets. And that was just to see if they are attentive during the task. We used pretty standard analysis for the imagery part. We went from the sensor space to source space by using MNE and DSPM. And we defined individual regions for each subject in the auditory cortex. And we extracted the time courses, source time courses from those areas. So these figures show the grand average signal from both conditions. So we see that in the auditory case, the amplitude is bigger, obviously, because it's auditory stimulus, and we look at the auditory cortex. Whereas in the visual stimulus presentation, if you look at the auditory cortex, the amplitude is much smaller and the signals are a bit more noisier. We can also see that in the auditory case, there are at least two main deflections. The gray areas here show the time windows, time points where these peaks were statistically significant. So we see first positive peak at about 40 milliseconds, and it's followed by a very big negative peak about 90 milliseconds time point. In the visual case, there was only one peak that was statistically significant, and it's a bit later at 125 milliseconds. So as we had two hemispheres and different tasks, we also, sorry, this is stuck. We also separately calculated the evoked responses for the noise task and letter task and left hemisphere and right hemisphere. So we can see that between those tasks and hemispheres, there are differences. But in our case, we were more interested in looking at the difference between auditory versus visual activity. So we use these grand average time courses for the modeling part. So then we were ready to start the modeling part. How we started, we were looking at the recent paper by Carmen Cole et al., who studied auditory evoked responses to 1 kilohertz sine wave stimuli. So that was our starting point, that it was almost the same stimulus. And they used HNN to model differences between hemispheres. So they found that there were differences between left and right hemisphere, but also contralateral and ipsilateral responses in relation to the presentation side of the auditory stimulus. So they were interested in looking at what could be causing those differences in the evoked responses. So they found by HNN that there were some specific parameters accounting for this. So for the right hemisphere dominance, they found that increasing the strength of the excitatory synaptic feedback in the right hemisphere, it accounted for the right hemisphere dominance. On the other hand, the contralateral dominance was related to the input latencies and number of cells that were used in the simulation. So as we were interested in a little bit different things, so we changed only the following parameters. So we were looking at the timing parameters when the feed-forward and feedback inputs arrive to the network, and their mean timing and the standard deviation. Also, we were looking at the effect of scaling factor for the simulated waveforms. Also, we limited this time window for the simulations a bit shorter. We wanted to look at the earlier effects as they are more pronounced. So for the auditory responses, we looked at the stimulus responses until 130 milliseconds and until 150 milliseconds for the visual response. So we took all the parameters from Carmen's model, and then we manually tuned them, those parameters I mentioned, to match our measured experimental MEG response. After that initial manual tuning, we further optimized the model parameters. We used Bayesian optimization approach that was implemented in GP minimize function. So we used MNE Python for doing this. So in this function, we gave bounds for the peak time and standard deviation for this auditory model. We have feed forward followed by a feedback. So this is the peak time bounds for the feed forward and standard deviation for the feed forward. Then for the feedback, the same peak time and standard deviation bounds. And for each iteration, we minimized the root mean square error between the simulated and measured signal. And we also included the optimizing the scaling factor inside the loop. So here we can see the simulation results. So the blue and orange show the experimental MEG responses, the grand average across the subjects. The gray area here shows the time window of interest, and the gray traces show the HNN simulations. And the lower row, it shows the feed forward and feedback input spike distributions and their timing. So if you look at the auditory case, there's initial brief feed forward input spike train, and it's followed by a feedback input that has more wider distribution. And in the visual case, we don't have the feed forward input, but we have feedback inputs with high standard deviation, and they come later in time compared with the auditory feedback. So these were our hypotheses. How we think it would be happening based on the previous literature on this topic. But how do we know that our hypothesis is the most optimal? So like with most of the computational modeling, there can be another set of parameters that could possibly create a similar kind of signal, or very close at least. So we built some alternative models that we compared our hypothetical models to show how our hypothesis relates to those. You can do it in many ways. You can have different sets of parameters that you vary what we did here, we varied the number of inputs to the models. So the first line shows the auditory models. So the outlined one is our hypothetical model. So we have one feed forward and followed by a feedback. Okay, now there are other combinations that could work as well. In the call paper, there was actually feed forward, feedback, another feed forward. So in our time window of interest, that actually looks pretty much the same. But we were focusing on the earlier parts, so we went with the simpler one here. Then we can ask, okay, what if we have only feedback input to the auditory model? How that would look like? So we can see that actually it fits the measured signal quite well, but now we miss the initial small positive deflection here. Then we tried, okay, what if we have only one feed forward? Do we need the feedback at all? Let's see how that looks like. And that didn't seem to work very well for our model. So this lacks feedback. So it's mostly if you look at the amplitude, it's mostly positive, and there is no feedback that would make it come more negative. So we, that's for the auditory part. And then this part is for the visual stimuli. Again, we could have visual stimulus with feed forward plus feedback. We could have visual stimulus with only feedback. That was our hypothesis. Or we could just have visual feed forward. So with the visual feed forward, the same thing happened. It didn't really work well. So it seems that we really need the feedback in the model. But then if you look at the visual with feedback or feed forward, feedback combined, we can see that actually it fits better. The feed forward feedback fits better to the measured signal. However, you also need to take into account the noise level of the signal. So if we look at this visual MEG signal, it has much more variance here in the baseline compared with the auditory one. So this could indicate that it just overfits. It fits to the noise. So we built a statistical analysis approach to look at the overfit and test which of these models we should choose. Auditory feedback or auditory feed forward feedback, and the same for visual. So I will go this through quite quickly because I see that I used already so much time. Maybe I jump to the conclusion slide. So we look at the variance in the experimental MEG distribution, and then we compared the simulation distribution to that. So the x-axis shows the error. So let's say we have this auditory feedback model, and then we compare it with feed forward plus feedback model. We see that we go to the left, so the error gets smaller. So the model gets better. At some point, there is a line where if you fit the model to the data, it might overfit. So it's not generalizable anymore. So we define this overfit limit to be 5% percentile of the MEG null distribution that reflects the variance in the MEG data. And we see that it's still within the limit, so we selected the auditory feed-forward feedback model. So it makes the model better, but it doesn't overfit. For the visual case, we see that adding the feed forward, it makes the fit better, but actually it seems that it overfits now because the RMS is smaller than the experimental variability. So we selected the simpler one. So in our study, we demonstrated the sensory-specific and cross-sensory MEG activity in auditory cortex, and we were able to explain the responses in terms of feed-forward and feedback inputs using HNN. And finally, I would want to say that I think HNN provides a very useful framework to go beyond the evoked responses that we always see. So we can really reach them with the animal works and really see what's going on underlying the responses. So when I started, I didn't have any prior experience with biophysical computational modeling. I heard about this framework from Matti Hämäläinen, who was still working in Martinez at that time. So I found very easy to start with the HNN GUI that allowed to experiment easily with the parameters. Later, Mainak introduced me to the Python version of that we used in our publication that allowed the scripting and being more adaptive in the workflow. So I want to thank all the HNN developers for your work, and thank you all for your attention.